So today we will learn the chapter 4, the epigenetics in fruit flies. Chapter 4 is comprised of the 6 subjects starting from the position effect variegation, PEV, and also poly-10 chromosome. And also we will learn the TRXG and there's PCG. After that we will learn about PV pathway. Finally, we will learn the dosage compensation mechanism. So in this slide, I will explain the fruit flies. So fruit fly has 4 pairs of chromosome, so 1 pair of sex chromosome and 3 pairs of autosome. For example, male has XY as a sex chromosome and female has XX as a sex chromosome. And also they have 3 pairs of autosome starting from chromosome number 2 and chromosome number 3 and chromosome number 4. And chromosome number 2 and number 3 and number 4. So their lifetime is that 30 days at 29 centigrade. Okay, and also their life cycle is the diploid status. So they are from the zygote to adult, they are zygote. Their diploid status 2n is 8. However, when they are in the gamete stage, for example, egg cell or sperm stage, they exhibit the haploid status. N is 4. Okay. So in this slide, I will explain the first subject, PEV, position effect variegations. So we learned this concept in chapter 1 briefly, but today we will see some details of post effect variegations. Okay, there is a very famous example. We already learned that allele W. Okay, so let's start from the allele large W or allele small w in Drosophila. For example, allele large W, this one is if the allele large W is expressed, it exhibits red pigment, and when the allele small w is expressed, it will exhibit the white pigment is like this. Okay, but allele large W is dominant than the small w. So in the case of when the genotype is large W, in this case, the eye color will be the red, and the genotype is large W and small w. In this case, is also the fruit fly will exhibit red eyes. But in the case of small w, it will exhibit white eyes like this. Okay. So when the allele large W is away from the heterochromatin, so it means that heterochromatin spreading cannot take place up to allele W position. So it means that allele W can be expressed, so it means that it will make the red eyes like this. Okay. So when the allele W is near heterochromatin region, so it means that heterochromatin spreading happens. So heterochromatin spreading changes allele large W position to heterochromatin regions like this. So it means that allele large W is inactivated, but allele large W also can be activated during some specific time point. So in these cases, they will have red eyes with white sectors like this. So this is the position. This is we learned that the position of allele large W, position effect variegation. Okay, so this is position effect variegation. So in this slide, we will learn the genes affecting PEB. So first of all, we will learn the SUVAR, suppressor of variegation. So there are several types of genes affecting PEB like this. This table summarizes the genes affecting the PEB. Okay, let me start the SUVAR. So SUVAR is suppressor of variegation. So there are several types of the SUVAR is here. Okay, so what is the suppressor of variegation? So suppressor of variegation means that suppress variegation when a mutation occurs in SUVAR. The first one is the SUVAR 3-9. Okay, so the main function of SUVAR 3/9 is their function is that H3K9 methyl transferase. So this gene is located in chromosome 3. Okay, so when a mutation occurs in SUVAR 3-9, so it means that there will be no H3K9 methylation. It means that there will be no heterochromatin spreading and formation. So it means that there will be no heterochromatin spreading and formation. So it means that it will suppress variegation. So this is the SUVAR, suppress of variegation. Another example is that SUVAR 2/5. So the main function of SUVAR 2/5 is HP1. So the main function of HP1 is it can recognize H3K9 methylation. So it will initiate heterochromatin formation. Okay. So this is in this slide I summarized the SUVAR. Especially the first one is the SUVAR 3-9 and second one is SUVAR 2/5. The important thing is that suppress variegation when a mutation occurs in SUVAR. So in the case of the SUVAR 3-9, so if a mutation occurs, so there will be no H3K9 methylation. It means that there will be no heterochromatin spreading and formation. Finally, so it will suppress variegation. Okay. So in this slide, I will explain the EVAR. So EVAR means that enhancer of variegation. This means that enhance variegation when a mutation occurs in EVAR. There are for example, this is the gene affecting PEB. So most of them is the SUVAR. In the case of there are two cases, the EVAR. The very famous one is the TRL. So TRL, the main function is they are a GAGA factor. So the function of a GAGA factor, so it recruits the chromatin remodeler. After that, it will make more open chromatin structures like this. After that, the RNA polymerases can be bind to this region. After that, transcription can be happens. So when a mutation occurs in TRL, so chromatins maintains heterochromatin status. So means that when their status in the heterochromatin, so it means that it will enhance variegation. Okay. So the EVAR means that enhancer of variegation. The very famous example is a TRL, is a GAGA factor. The main function of GAGA factor is makes more open chromatin structure. But when mutation occur in the GAGA factor or TRL, the chromatin maintains heterochromatin status. It means that heterochromatin status is enhanced variegation. This slide, I will explain the second subject, polyten chromosome in Drosophila salivarigrans. So what is polyten chromosome? So polyten chromosome is that the size is very big. Okay, so usually the very big size chromosome is found in rapidly developing tissue and organ in fruit flies. Especially this polyten chromosome easily found in the salivary glands. Okay, so this is fruit flies. This is fruit fly larvae. Okay, so fruit fly larvae is composed of upper region and the down region. So if we separate the upper and down region, you can see the salivary gland. So what's the main function of salivary glands? So it secretes alpha amylase. The alpha amylase is an enzyme that breaks down the starch into maltose and glucose. Okay, so this is the salivary glands. So if you magnify the salivary gland, so this is salivary glands. We see here most of cases, most of space, the nucleus occupies the space, and this is the polyten chromosome. So polyten chromosome is 100 and 200 times larger than normal chromosome. So this is the normal mitotic chromosome at fruit flies. So it means that this is the polyten chromosome is 100 and 200 times larger than normal chromosome. Okay. So in this slide, I will explain the detailed structural information of polyten chromosome. So this is the normal chromosome structure in the fruit flies. They are composed of the 1 pair of sex chromosome and 3 pairs of the autosome, starting from chromosome 2 and chromosome 3 and chromosome 4. Okay. Okay, how about in polyten chromosome? Okay, here we can see that chromosome 2 left arm, so they have left arm, and also this is chromosome 2 right arm. This is right arm. How about here? We can see that chromosome 3 left arm, so this is left arm, and that there is the chromosome 3 right arm is here. So right arm is located here. So actually, and also there is a chromosome 4. So initially chromosome 4 is the size is very small, like this. So in here they have the left and right arm together. Okay, here. Okay. How about chromosome X? So this is the chromosome X. This is X chromosome. Okay, here. How about the Y chromosome? So, for example, here Y chromosome is not visualized as they are completely fused with the chromocenter. So this is the chromocenter. So Y chromosome is completely fused with the chromocenter. So Y chromosome is located in the chromocenter region. Okay. Okay. So in this slide, I will explain some details, information of polyten chromosome, especially focus on heterochromatin and euchromatin. So previously we learned that heterochromatin is the more condensed region and also stains darker. Okay, so usually located in the telomere region and also centromere region. How about euchromatin? So euchromatin is less condensed and also stains right is like this. In polyten chromosome, this is polyten chromosome. If we stain the polyten chromosome, so we will see some dark region, band region, and also light, very light region, like very light region. Okay, so if we magnify this region, so band region is very condensed DNA, so that's why it stains darker. So this region is usually the heterochromatin region in polyten chromosome. How about the interband region? So in this region, the DNA is very relaxed. Means that maybe chromatin structure will be very relaxed. So it stains very light like this. Okay, so this region is the euchromatin region. Okay. So in this slide, I will explain the third subject, 3 drugs group proteins, TRXG proteins. So what's the role, main function of TRXG? So this protein determines segment identity. So this is the fruit flies larvae. So they have several segments. So TRXG proteins determines segmental identity from anterior to posterior. Okay. So and also their main function is to maintain the active status of gene expression. So what's the meaning of the maintain the active status of gene expression? So let me introduce one example. For example, to make the T3 rags, okay. So in the case of T3 rag, okay. So UBX gene should be expressed in this segment. Okay. So in the case of the UBX gene, usually the TRXG proteins maintains the UBX gene expression in T3 segment. Okay, that's why T3 leg can be made like this. Okay. How about in the T2 legs? To make the T2 leg, okay, to make the T2 leg, antenna P gene should be maintained in T2 segment. Okay, in these cases, also TRXG maintains the antenna P expression in T2 segment. How about the T1 leg? This is T1 leg. So to make the T1 leg, so SCR protein, SCR gene should be maintained in the T1 segment. Okay, so usually the TRXG is the main function is maintain the active status of the gene expression. Okay. So TRXG is composed of 3 different kinds of proteins. The first one is the chromatin remodeling TRXG protein, especially the ATP-dependent chromatin remodeler. Okay. Second one is the histone-modifying TRXG proteins, and also especially the histone methyltransferase, and also third one is the DNA binding TRXG proteins. Okay, so let me focus on the chromatin remodeling TRXG proteins. So the very famous example of the ATP-dependent chromatin remodeler is SNF complex. So this chromatin remodeling proteins alter position of a nucleosome along the DNA, so it renders DNA is accessible. So there are several cases. So it renders DNA accessible. So it can maintain the active status of gene expression. And also there are another type is the histone modifying TRXG protein, and also there are DNA binding TRXG protein, but in this slide I will not cover in details. Okay, so in this slide I will explain the fourth subject, PCG. The full name is polychrome gene. Okay, PCG is that also determine the segmental identity from anterior to posterior. Their specific function is to suppress the active status of gene expression. So this specific function is totally opposite to a TRXG function. Okay, TRXG is also determines segmental identity from anterior to posterior. But the specific function is maintain the active status of gene expression. However, PCG is suppress the active status of gene expression. So let me introduce one example. So we already learned that to make the T3 reg, to make the T3 reg, UBX gene should be expressed in the T3 segment. Okay, so at that case is TRXG maintains the active status of UBX expression. But however, except for T3 segment in T2, T1, and also the other segment, so UBX expression should be suppressed. So in this case, polychrome G protein is suppressed the active status of UBX expression in this segment and also in this segment. Okay, so through this functioning, so it also can determine segmental identity. So it will make the T3 regs like this. So determine segment identity from anterior to posterior. Also usually they suppress the active status of gene expression. Okay, how about in the T2 segment? So T2 segment, to make the T2 regs, okay, TRX in that case, TRXG maintains the active status of the antenna P expression in T2 segment, but except for T2 segment, the other region, the expression of the antenna P should be suppressed. At that case, the PCG suppressed the active status of the antenna P expression in the other segment. Okay. So PCG is composed of two types of the protein. One is PRC1, the other one is PRC2. Then PRC1 we will learn in this slide, and PRC2 we will learn in the next slide. The main function of PRC1 is to suppress the active status of gene expression. So PRC1 is composed of the PC and SCE and PSC and also PH. And table 4.3 summarized the each component of the PRC1 complex subunit. Okay. Let's briefly see each protein's function. Okay. First one is the PC. PC is the main function is to recognize H3K27 methylation. The second one is SCE. The SCE is that the full name is SDE and slash ring 1. This protein is generate H2A K119 ubiquitination, especially H2A histone. Especially histone H2A histone is composed of several amino acids, especially 19th location. K is lysine. Lysine is located there. Especially the SDE ring 1 generate ubiquitination of the H2A K119. So this ubiquitination promote the methylation of another histone H3. So finally, after the histone H3 methylation, so it can suppress the active status of gene expression. How about PSC? So PSC is also can generate H2A ubiquitination. Okay, and so we will not cover the pH here. Okay, pH is also the subunit of PRC1. Okay, but we will not cover in details. How about the PRC2? The main function of PRC2 is also suppress the active status of gene expression. So PRC2 is composed of the several protein. Okay, starting from the EZ and the ESC and NRUF55, and also you can see the other proteins here. Okay, let's start from the EZ. Okay, EZ. So this is the PRC2 in Drosophila. So EZ is located here. So main function of EZ is the H3K27 methyltransferase. So this EZ is H3K27 methyltransferase. Okay, how about ESC? So ESC is recognized. EZ is the methyltransferase. ESC can recognize H3K27 methylation. How about NRUF55? So NRUF55, so this is the NRUF55. Okay, so NRUF55 is located here. The main function is it just bind to RNA. Okay. So also there are other subunit. There are other protein in the PRC2 subunit, but we will not cover in details. Okay. So the main function of PRC2 is suppress the active status of gene expression. So main function is it can cause facultative heterochromatin in higher eukaryote. So PRC2, they are composed of H3K27 methyltransferase, and also ESC can recognize H3K27 methylation. So these two protein is the main function to cause. The main function is to cause facultative heterochromatin in higher eukaryote, not the constitutive heterochromatin. So this PRC2 make the facultative heterochromatin, means that this facultative heterochromatin can be changed to easily changed to euchromatin. And also composition of PRC2 in porpoise, Drosophila, and also human, the mammalian, is nearly similar to each other. Okay. So in this slide, I will explain the facultative heterochromatin formation by PRC1 and PRC2. So there are three different pathways. So the two is the PRE dependent pathway. This is also PRE dependent pathway. And finally, there is the PRE independent pathway. So let me start PRE dependent pathway. Okay. So in the DNA sequence region, so there is a PRE region exists. PRE region is a PCG response element. The DNA sequence is the 5', starting from 5', C, cytosine, and N means that adenine, thymine, guanine, cytosine can be here. The C, N, guanine, C, C, A, T, N, N means that adenine, thymine, guanine, cytosine can be here. N, G is like this. So usually the 4RC, the transcription factor, bind to this region. So this is the 4RC. 4RC is transcription factor. So this pathway is transcription factor dependent. Okay, this factor bind to the PRE region. After that, it will recruit PRC1 and PRC2. So it will recruit PRC2 and PRC1 to 4RC. Except that PRC2 induced H3K27 methylation. So because the PRC2 has the EZ subunit, okay, this subunit, its main function is H3K27 methylation. H3K27 means that histone 3. So histone 3 is composed of several amino acids. 27th location, there is K means that lysine. The lysine. So lysine. So this lysine location can be methylated. Okay. So PRC2 induces H3K27 methylation in PRE region. After that, PRC1 recognized H3K27 methylation in PRE region. Actually, the PRC1 has PC region. So this can recognize the H3K27 methylation. After that, it will make the chromatin compaction, and also finally it will make the facultative heterochromatin. Okay. Second pathway is also PRE dependent pathway. So usually starting from the PRE region is methylated. Okay. Some of the PRE region is methylated. So methylation in the PRE region recruit PRC2 and PRC1. So PRC2 has EZ subunit. So EZ subunit, especially this subunit, induces H3K27 methylation in the PRE region. And after that, PRC1 recognized H3K27 methylation in PRE region. So it will make the chromatin compaction, and finally it will make the facultative heterochromatin. Okay, so finally there is another pathway, PRE independent pathway. So previous one is only happened in the PRE region, and also the first pathway is also occurs in PRE region. But so this PRE independent pathway, it means that so it will be occurs everywhere. Okay, especially the PRC2 bind to the RNA, some kind of the non-coding RNAs. Okay, why PRC2 can bind to the RNA? Why PRC2 can bind to the RNA? Because the PRC2 has NURF56 the proteins. NURF56 protein. The main function is this is RNA binding. Okay, so PRC2 bind to the non-coding RNA, especially non-coding RNA. After that, PRC1 also can bind to this complex. So after PRC1 binding, and so after PRC2 binding, and PRC2 has the NURF56 the proteins. So this subunit bind to the RNA. After that, PRC1 can bind to this complex. Complex means that PRC2 and the RNA, this complex. Okay, so finally these complex bind to the DNA, some specific DNA, some DNA. After that, PRC2 induce H3K27 methylation, and the PRC1 recognized H3K27 methylation. So finally, the chromatin compaction happens. So it will make the facultative heterochromatin formation. Okay. So this is the PRE independent pathway. So PRE dependent pathway occurs only in the region which has PRE, the element, PRE sequence, but PRE independent pathway can be occurs in everywhere. Okay. So it bind first the PRC2 bind to the RNA, and after that it will recruit PRC1. So finally this complex bind to some DNA region. So after that PRC2 and PRC1 they do some specific function. Okay. So finally, previous slide I just mentioned the PRC2 and PRC1, but now in this slide I will explain some detailed mechanism of facultative heterochromatin formation by PRC1 and by PRC2. So usually the PRC2 induces the H3K27 methylation, but especially the EGIT. EGIT has the PRC2 some specific subunit. EGIT they can do the induces H3K27 methylation, and also PRC2 has the ESC subunit. So this ESC subunit also recognized H3K27 methylation, and also PRC1 also especially in PC region is the recognized H3K27 methylation. So finally, so PRC2 induces methylation, H3K27 methylation, and also PRC2 also has some ESC domain. It also can recognize the H3K27 methylation, and also PRC1 also can recognize H3K27 methylation. So finally, it will make the chromatin compaction, and also this region will be the facultative heterochromatin. So this is we run the PRC2 and PRC1. So each subunit has some specific function. So when we purified each subunit, so in the PRC2 had several subunits, and PRC1 had also several subunits. So when we purify each subunit and tested whether is it shows that H3K27 methyl transferase activity, but we found that each subunit they did not show their own the function. So when we purify just this complex or this PRC1 complex, so at that we found that when EZ or ESC is belong to this PRC2 complex, and also PC and another their subunit is belong to the PRC1 complex. So when they existed in complex and exhibit H3K27 methyl transferase activity, and also ESC can recognize H3K27 methylation. Okay. So we learned that some specific mechanism of facultative heterochromatin formation by PRC1 and PRC2. Okay. So in this slide, I will explain the fifth subject, suppression of gene expression by RNA, especially the PV pathway. So previously, we learned that the RNAi dependent heterochromatin formation. So at that time, we found that ARG1 initiated RNAi dependent heterochromatin formation in Fusionist. How about in fruit flies? Who initiated the suppression of gene expression by RNAi? So we found that ARG1 ortholog means that one of the homologous gene sequence found in different species. For example, in fruit flies, so we found that PV or AUV and ARG1 and ARG2 and ARG3, they did similar function as the ARG1. So especially the PV did a very important role in the suppression of gene expression by RNA. Okay, so what is PV? So PV is full name is P element induced Whimsy testis. Okay, so PV is initially understood only occurs in germline cell, especially testis or the egg. Because the reason why the problem with PV causes infertility in the male fruit fly or female fruit flies. But we also found that PV is also works in the somatic cells. So it means that PV works in the germline cell and also works in somatic cell. Okay. So in this slide, I will explain the PV interacting RNAs. And the short name is piRNA. So piRNA is a small long-coding RNA. So in small long-coding RNA, there are three types of the small long-coding RNA exists. piRNA and the siRNA and the microRNA, microRNA. And the matured piRNA size is about 26 and 31 nucleotide. Okay, usually the piRNA, after piRNA usually form RNA protein complexes through interactions with PV. So PV interact with the piRNA, so they will make the RNA protein complexes. So until now on, 100,000 piRNA was identified. How about microRNA? So until now on, only 500 is identified in microRNA. So main function is piRNA is usually the piRNA is produced during egg cell formation, not sperm cells formation, during egg cell formation. After that, the piRNA after fertilization, so piRNA transferred to the zygote, and the main function of piRNA is the piRNA spreads the expression of a transposable element. So in our human genome, we have the transposable element. The main function of transposable element is this transposable element inserted in anywhere in our genome. So it will disrupt our gene function. The very famous example of the effect of transposable element is cone color changes in the cone kernel color changes in the cone. Okay, so actually the piRNA is evolved to protect our human genome. So because the transposable element can disrupt all of the human gene, so piRNA main function is protect our genome, because piRNA can suppress the expression of the transposable element. So in this slide, I will explain the piRNA and post-transcriptional gene silencing. So this mechanism is composed of a primary piRNA pathway and secondary piRNA pathway. So in this slide, I will explain the primary piRNA pathway. So this is the piRNA DNA, which is composed of the piRNA cluster. So inside the nucleus, so piRNA precursor transcript was synthesized during the egg cell formation. Not sperm cell, just the egg cell formation. During egg cell formation, just the piRNA precursor transcriptor, which is composed of sense transcriptor and antisense transcriptor. Okay, after this piRNA precursor transcriptor has been synthesized, these were transported to the cytoplasm. So this is a cytoplasm. After that, Zik protein is bind to piRNA precursor transcriptor, especially the antisense transcriptor. After that, it will chop it, make it cleaves the transcriptor and make it intermediated RNA. After that, PV or AUB is bind to the one of the intermediate RNA. After that, trimmer is to induce this 3' trimming of the intermediate RNA. And finally, the HANT, so induce 3' methylation. So what's the function of the main function of trimmer and HANT? So after 3' is trimmed, or after 3' become the methylated, so the antisense, this RNA will be very stable. It's not degraded anymore. Okay, so this is the primary piRNA pathway, starting from piRNA cluster. After post piRNA precursor transcript was synthesized during the egg cell formation. After that, this precursor was transported into the cytoplasm, and Zik bind to the antisense transcriptor and made the intermediate RNA. After GV or PV or the AUB is bind to the intermediate the RNA. After trimer did 3' trimming, and also hand did 3' methylation. So after finish this procedure, so this the RNA will be very stable. They will not be degraded. They will not be easily degraded. So in this slide, I will explain the secondary piRNA pathway. So the antisense piRNA with AUB. So antisense piRNA with AUB is bind to sense transcriptor of the transposon. So in the cytoplasm, there is the sense transcriptor of transposon, sense and antisense transcriptor of transposon will be existed. Okay, so first the antisense RNA piRNA with AUB is bind to the sense transcriptor of transposon and cleaves the transcriptor. So first, antisense piRNA with AUB binds to the sense transcriptor of transposon and cleaves the transcriptor. Okay, after that, this part will be the 5' fragment, and this part will be the 3' fragment. So this is so after that A goes 3, R goes 3 is bind to the 3' fragment. After that, trimer induces 3' trimming, and also hand induces the 3' methylation. Okay, so this is first part of secondary piRNA pathway. Okay, in the next slide, I will explain the second part of secondary piRNA pathway. So after that, now this RGO3 is bind to the sense piRNA. Okay, after that, sense piRNA with RGO3 bind to antisense transcript of transposon. Okay, after that, it will cleave the transcriptor. Okay, so this part will be the 5' fragment, and this part will be the 3' fragment, and 3' fragment. After that, AUB or PV or AUB recognize the 3' fragment. After that, trimer induces 3' methylation. Trimer may induce 3' methylation. After that, hand induces the 3' methylation. So in these cases, after that, the antisense piRNA with AUB bind to the sense transcriptor of transposon. So and cleaves the transcriptor. Okay, after that, this pathway will be repeated. So it means that sense transcriptor of transposon or antisense transcriptor of transposon was actually initially transcribed in the nucleus. After that, this sense and antisense transcriptor of transposon can be transported into the cytoplasm. So after that, secondary piRNA pathway cleaves nearly all of sense and antisense transcriptor of transposon. So it means that after post-transcription, transcription happens. After that, the sense and antisense is transported into the cytoplasm. So the sense transcriptor of transposon and antisense transcriptor of transposon can be degraded. So this is recorded as a post-transcriptional gene silencing. So finally, it will induce PTGS. Okay. Chromosome X in the Drosophila. Okay, so Flamengo piRNA is located near the centromere region of the chromosome X. So this region is heterochromatin region. Okay, so this Flamengo transcript is synthesized. After that, it will make the sense and antisense transcript. After that, this transcript, after that, okay. However, this Flamengo has the gem fragment. So originally, full-length gem is the retrotransposon. This gene is also located in chromosome X. Okay, but this Flamengo transcriptor has a small part of the gem gene. So we call it as a gem fragment. Okay. So after that, this transcriptor is transported into the cytoplasm. After that, piRNA biosynthesis procedures happen. So we already learned that Z bind to the transcriptor. Also, PV loading happens. After that, trimer and hand, they did their special function. Okay, so after that, this PV with piRNA is imported into nucleus. So after that, this piRNA PV, this one bind to the gem transcript. So this binding decrease the synthesis of this gem transcript. Okay, so it means that because the gem transcript is not synthesized, so it induced transcriptional gene silencing. Okay, so maybe the possible reason is that PV and piRNA with the PV is bind to this region, so they will not be dissociated from this region. So maybe polymerase 2 cannot initiate the second round transcription, and third round cannot initiate third round in the transcription. Okay, previously we also learned that TGS. So at that time is the Schreck complex is inhibit RNA polymerase binding to the promoter region. So it reduced it induced decreased RNA synthesis, and after it induces gene silencing. But in these cases, piRNA with PV bind to gem transcriptor. So these bindings decrease the synthesis of gem transcriptor. So this is induced transcriptional gene silencing. Okay. So in this slide, I will explain the final subject, sex chromosome in fruit flies. So I will explain the dosage compensation mechanism. Okay, so dosage compensation mechanism in fruit flies is different from the dosage compensation mechanism in mammalians. So let me start dosage compensation in fruit flies. For example, in fruit flies, male has sex chromosome, XY sex chromosome. How about female? Female has XX chromosome. How about in the mammalian? Mammalian also has the male has XY sex chromosome and female has XX sex chromosome. Okay, but in the case of the fruit flies, dosage compensation mechanism happen in the male. Okay, especially in this X chromosome. Okay, here, so fruit flies X chromosome, fruit flies male's X chromosome. This X chromosome can be transcribed 2 times faster. So it means that the amount of transcript synthesized on X chromosome will be doubled. That's why the amount of transcript synthesized on X in male will be as the same as the amount of transcript synthesized on XX in female. So this is the dosage compensation mechanism in fruit flies. How about the mammalian? So mammalian, the dosage compensation mechanism happen in the female. So female has the XX chromosome. So in this case, the random inactivation of one of XX happens. So one of XX chromosome can be randomly inactivated. So that's why only X chromosome, one X chromosome can be transcribed. So this is the dosage compensation mechanism in mammalian. So in this slide, I will explain the example of dosage compensation in fruit flies. So in 1930s, Dr. Mueller identifies that allele W, which encodes eye color, locates on X chromosome. So he also identified that female has 1 and 2 allele W's, but male only has 1 allele W. But he found that there was no big difference in red color density between female and the male. So this is the one example of the dosage compensation. So we will learn some detailed information in next slide. So in this slide, I will explain the dosage compensation mechanism in fruit fly, especially focused on the dosage compensation complex, DCC. This DCC is existed in the male X chromosome. However, female X chromosome, they have some part of the DCC complex, but they don't have a full complex. Okay, let me start from the complex subunit. First one is MOF proteins. So this is the MOF protein. MOF is the H4K16 acetyltransferase. So histone H4 is composed of the several amino acids, especially 16th amino acid is lysine. So MOF can induce the acetylation, so it will make the H4K16 acetylation. So we already learned in chapter 1. So histone acetylation, when histone acetylation happens, so heterochromatin can be changed to the euchromatin region. Okay. The second subunit is the MLE. So MLE, the main function is RNA helicase. So RNA helicase, this is RNA helicase. So RNA helicase is unwinding or annealing the RNA molecules. So after that, these RNA molecules, the ribosome can be easily bind to the RNA molecules. After that, the translation initiation can be easily happens. Okay, the second subunit is the MSL1. So MSL1 is also component of histone acetyltransferase complex. Okay, and also they are composed of the MSL2 and 3. So MSL2 is the only expressed in the, we can only find in the male X chromosome, but in the female X chromosome we cannot find the MSL2 subunit. And also DCC complex has the also have the MSL2 and MSL3. Okay, and also they have the LOX ROX1 and ROX2. ROX1 and ROX2. So this is the non-coding RNA. So this ROX1 and ROX2 is only existed in the male X chromosome, but in the female X chromosome, they don't have the ROX1 and ROX2 non-coding RNA. The main function of the ROX1 and ROX2 is recruit, so MOF and also MLE and also MLX complex together. So okay, so apt TCC complex is they function each other. After that, TCC complex can transcribe X chromosome 2 times faster. Okay. So in this slide, I will explain the DCC binding mechanism in male X chromosome. There are 4 different models, starting from the model 1 is this DCC complex is bind to the one region. After that, it will induce long range spreading. So this is X chromosome. So one binding, after that it will induce long range spreading. So model 2 is that limited number of the spreading, binding. After that, it will induce the local spreading. So this DCC is bind to this region, this region, and also it can induce local spreading. Okay, this is the second model. The third model is there is the limited number of recruitment and without spreading. And model 4 is that high density of recruitment site and without spreading. So nowadays people found that model 4 is right. So people observed the high density of DCC binding site on the particular sequence. DCC is bound to the CES, chromosome entry site, CES site. So DCC can be bound to this CES site. Except DCC bind to this region, it will increase the transcription rate 2 times. Okay. So today we learned the chapter 4, the epigenetics in fruit flies. First we focused on PEB, and also we learned the polyethylene chromosome, and also we learned the TRXG and the PCG. And also we learned about the PV pathway. Finally, we learned sex chromosome in fruit fly, especially focused on dosage compensation mechanism. So today we will finish the class.